start the live streaming on YouTube.
Okay, sir.
Okay, great. So welcome everybody to this panel discussion, which is going to be on the ethics of genome editing. And it really was something which, when we started organizing the conference, Sudarshan and I, we really thought it's important to also put together such a panel discussion, because when you start thinking about genome editing, immediately ethical concerns, what are the regulations? These are things that come to mind. And we've actually seen over the past 2 days of the conference that people have posted these questions and posed these questions to some of the speakers. So it's really great that we could organize this panel discussion, and we're really a big thanks to all of the panelists that they could be here today and share their very valuable insights into what is happening in this area in India specifically, because as we know, many of these regulations are country specific. So it is important for us to know what is happening in India, and also to some extent, what is the international context of it. So I will not take any more of your time. So I will hand it over to the moderator of the panel discussion, Dr. Rohan Kamath. So Dr. Rohan is currently working with Immunil Therapeutics, but prior to that, he has experience where he had a startup company that worked with CRISPR-Cas editing. So it really is quite relevant for us to have him here, and he will be able to moderate the discussion, and he will take over. So welcome, Dr. Rohan, and a big, very warm welcome to all of the panelists. So thank you very much.
Great. Thanks a lot, Shruti. Bear with me for my bad voice that I'm just reeling out of a bad sore throat. So good evening, everybody, and welcome to this panel discussion on the ethics of genome editing. Something that always crops up whenever you mention something that tries to alter the path of nature, as a lot of people call it. So first of all, thanks a lot for the organizers for getting this fantastic talent pool in this meeting. Now, we also have some key opinion leaders in their respective domains on the panel, and I would like them to introduce themselves. I will just take your name, and please introduce yourself to the whole meeting. We can start with Professor Srinivasan from Johns Hopkins, please.
Hi, I am Professor Chandrasekharan from Johns Hopkins. Actually, I'm a Professor Emeritus. I took my retirement in 2020. I'm spending more time I'm going to spend more time in India, especially helping my brother's small biotech company, advising scientists. Okay, and I'll also be working with my colleagues at IISc and of course IBM. Okay. It's a pleasure to be on this panel, and I'll stop there.
Thanks a lot, Professor Chandrasekharan. It's great to have you back in India. We need a lot of people like you here to continue with the great science that happens around. Thanks a lot. So moving over to Dr. Shambhavi. I know Shambhavi for quite some time now, and I'm not going to call her Dr. Shambhavi, but just simply Shambhavi. Shambhavi from THSTI think tank, and she also has a startup called CloudCard. I'll let her introduce herself. Shambhavi,
I was going to say, please don't call me Dr. Shambhavi. So thanks for saying that up front. Hi, everyone. I'm Shambhavi. As Rohan mentioned, I work at a think tank called THSTI. I've been studying gene editing policies in India since 2017. And I also have co-founded CloudCreate, which is into supply chain for life science products in India. I'll just stop there. Thanks, man.
Thanks a lot, Shambhavi. Moving over to Professor Vishal Rao from Healthcare Global. Professor Vishal Rao, please introduce yourself to the team out here.
Hi, very good evening. I'm sorry, there's some issue with my camera setting. I'm trying to turn it on. I am Dr. Vishal Rao. I'm a surgical oncologist and also the Dean of Academics for Healthcare Global. We are a chain of cancer centers. We are about 23 cancer centers across the globe right now. And our primary focus is in oncology. We also have our vertical of genomics, where we very actively work on cancer care at early detection, diagnostic, prognostication. And I'm a member of the Karnataka's COVID task force for the Genomic Surveillance Committee.
Great. Thanks a lot, Dr. Vishal. Great to have you here today. Thanks a lot for taking out time for this meeting. We'll move on to Dr. Viva Ahuja from the BCIL Delhi. Dr. Viva, please, it will be great to hear you.
Yes. Thank you so much. And thank you for inviting me to be part of this panel. I am the Chief General Manager of Biotech Consortium India Limited, which is a company which was promoted by the Department of Biotechnology to accelerate commercialization of biotechnology. So in the process of commercialization, regulatory issues are very, very important, and they are, in all biotech products. So BCIL has been working in the biosafety issues related to genetically modified organisms for almost more than 25 years, and very active in the early approval processes working with the government and with the developers. As the gene editing technologies have emerged now, we are actively engaged in that, associated with the process of the development of the regulations, as well as creating a lot of awareness and sensitization, reading about and creating webinars on how the situation is moving in other countries. We've done several with the CG system and all, but our focus, I would share with you here, has been more with the gene editing in agriculture rather than the medical. So I would be sharing with you the experience which is now there that we have during the course of course, the panel discussion, more related to the plants area rather than the medical area. Thank you so much.
Thanks a lot, Dr. Viva, and I think that's a really important point and point of discussion, because agriculture, which becomes the backbone of India, really needs the policies which can help us use a powerful technique like genome editing and ensure that we do not cross our boundaries, but at the same time take the most benefit out of it. So thanks a lot. We'll listen more about that soon. Do we have Professor Manisha Inamdar online as yet?
Doesn't look like it, Rohan. So
yeah,
just
okay.
I can quickly update people. There was this, I'm sure many of you are aware of the disastrous flooding that happened in JNCSR in Bangalore earlier this week. So Professor Manisha said she would try to make it depending on how the situation is. So I guess she's probably quite preoccupied with that. So we do thank her for agreeing to be on the panel, but unfortunately she might not be able to make it today. So you can carry on.
Yeah, thanks a lot, Shruti. Indeed, a very troublesome rain which caused a lot of havoc in the north of Bangalore. Great. So we've heard some fantastic talks in the meeting with respect to various aspects, and
You've gone on mute.
oops, I'm really sorry. Thanks a lot. Standard Zoom issues. So yeah, I was saying that we've heard a lot of fantastic talks in this meeting, right, from various aspects of genome editing, ranging from the new tech, the new Cas9s, the new CRISPR methods of designing gRNAs, to diagnostics, therapeutics, and then the evergreen agriculture, as I call it. What we would like to do is we would like to hear from each and every of the panelists out here about how genome editing has impacted or played a role in their field, in their experience, or you feel that it can make a big difference to your field. And of course, that's the good part about it. But we'd also want to hear that when you say that I think that this will be great, we would also like to understand how it may pose a risk, if at all, because that's very critical considering this panel where, we address the, it's a seesaw where we are balancing the risks and the benefits. So I'll start with Shambhavi, since you've been looking at policy for a long time. We've exchanged notes on multiple platforms, and we have a lot of similar thoughts. I would like to hear about what has been your experience with respect to genome editing and its policies since you've started engaging with us.
Right.
Thank you.
Thanks, Ron. So I'll keep the advantage to a very generic level because I think people who are experts in particular fields are better posed to speak about those. My one take on the one thing I'm very excited about genome editing is actually fundamental R&D, just increasing knowledge of ourselves, of plants, of animals, right? Using gene editing in fundamental R&D, I think is something that is really low hanging. And the more we converse on ethics and issues, my fear is that if we lose out on that one particular application, that's going to cause us the maximum harm. The other, I'm sure others will speak about health security, nutritional security, etc. So I won't go deep into that. I'll talk about risks that I see as a policy analyst. So obviously there's fear of this whole super soldier bioweapon kind of thing, and that narrative has been capturing people's imagination, right? And the fear for me as a policy analyst is that we go into that territory where people are too scared because there is no clear policy. When people have been hearing about these CRISPR babies, is China making super soldiers? There was this news about one of the companies in China tying up with a genetic testing lab to try and get a lot of samples analyzed on what are the best genes that contribute to muscle strength, etc. Right? And my fear is that if we don't have clear policies from the beginning, that narrative captures imagination, and we don't get to the benefits of using gene editing. So one big risk for me is unclear legislation. The second is when we have policies, but we have copy pasted them or imported them from outside, and they have not been contextualized for India. For example, is germline gene editing unethical? Who gets to decide that in India? Is it a committee within DBT or ICMR, or whether the Ministry of Health and Family Welfare, or do we have a process by itself in which we figure out what do Indians actually think about ethics of germline gene editing, right? Then once we have figured that out and we figure out that, hey, maybe we should be doing this, are we then saying that, look, these are the US laws, these are the UK laws, this is what we should do? Or are we saying this is the thalassemia burden in the US, these are the other kind of services that the US offers to someone who has thalassemia? For example, ramps in schools, something very simple, but we can't offer that in our government schools to our children. Could then gene editing be something that we can offer, right? We develop because of that, because our context is different. And the third is this blanket bucketing of ethics of regulation for gene editing instead of looking at particular applications. Germline gene editing is not the same as gene editing in mosquitoes, not the same as plants. So how do we now go about categorizing and figuring out good sort of regulation where we can capture the benefits and kind of try and monitor the risks? Thanks, Ro.
Thank you, Shambhavi. Absolutely agree. Policies need to come early in time. Copy pasting is not a great option. Absolutely agree. Okay, moving over. Professor Chandrasekharan, in your experience, you've been doing science for a very, very long time, and you've seen the advent of I would say genetic engineering, gene therapy, genome editing. What is your opinion about these policies, and especially when it comes to India? Because you've been in the US, from India, you've been to the US, now you're back here. How do you think we can learn from the US? And any comments about how Shambhavi says that, yes, we should not be copy pasting policies if at all, but we should be careful. We should be crafting out something for India. Will be great to hear your views about this.
Present you my views on the subject. First, I want to address the technology itself. What are the challenges facing genome editing? The technologies are not perfect. Actually, my lab worked on zinc finger nucleases, which is prior to CRISPR. Most of the young people in your audience probably never even heard of it. Then there were talents, then came the CRISPR. But all the three technologies have problems. the most important concern is off-target effects, right? They not necessarily cleave on target, they can cleave outside, okay, away from the target sites, which could pose a problem. So if you take a cut at an essential gene, you will mutate that gene. So that you have to keep in mind. The second is the efficiency of cleavage. They are not very high. They are still very low. So these have to be improved. Okay. Keeping those in mind, just thinking about the diseases. When you have an autosomal recessive disorder, right, both genes have to be mutated for you to have the disease. So in that case, all you need to do is correct one of the alleles in enough cells. If you can do that, there will be a therapeutic effect. Okay, that can be easily done. But there are diseases which are autosomal dominant disorders where just a defect in one allele will result in the disease. There, if you want to have a therapeutic effect, you have to correct both of them, both alleles, both copies of the genes. That's very difficult to do, and the efficiency is very low. So the other thing I'll say is how safe is delivery. Some people are now trying to do gene editing in vivo, essentially systemically deliver the reagents and hopefully go to the right target cells and correct them. Again, those are not very efficient, okay? So keeping all this in mind, so the technology has to advance pretty far to be really very useful in clinic, okay? Now bringing back, I'm, like you said, I am from US, so the US policy is right now they approve only somatic gene editing. They don't allow germline gene editing. It's obviously very simple for a simple reason. If you edit the germline cells, you are going to introduce mutations, and that mutation is going to be carried throughout the progeny. And if, like I said, if you have off-target cleavage on an essential gene, and that leads to a disease, that will be carried out all through your future progeny. So USA has banned germline editing, okay? Somatic cell is fine, because here most of the ideas are, right now, because the efficiencies are low, you isolate the cell. They do it ex vivo. You isolate your own cells, do the treatment ex vivo, amplify the cells, then put the cells back into your body for autologous transplantation. That's what most people are doing now. Most companies are concentrating now, and most of them are addressed to blood disorders where you can replace the cells. Okay, doing like cystic fibrosis is very difficult to do, because again you have to deliver the reagent, get sufficient cells corrected, and that, and it's the lung cells, it's very hard to do right now. So that's all far-fetched. Then the other thing I want to point out, even if everything is perfect, from point of India, patient treatment cost. Right now, the calculated cost, estimated cost is, let's say, $1 million per treatment. I think it's $10 lakh per treatment. Okay, that nobody can afford that. So the costs have to come down for it to be really useful. The other thing, with regard to genome editing of human embryos, my view is eventually this will happen. Once the technology is perfected, those things will happen. But right now, you don't do it, okay? You wait for the technology to advance sufficiently and it's safe, then you apply these. And you can't, especially you can't apply them when you have PGD available to you. that is prenatal gestational diagnostic, where you can already test your zygote. If there is a mutation, then you don't make it proceed to completion of the baby. So you can, these are safer techniques compared to genome editing. So when they are available, why do you need genome editing? Okay, so that's the other thing. But like I said, in USA, which is like society like ours, which is wealth driven, everybody wants the latest technology. So that's why I'm saying it will eventually happen because the patients want it. Okay, but we are not there yet. I wouldn't be afraid of designer babies. Okay, they are a long way off. Okay, but I won't allow anything for cosmetic performance, like somebody was saying, muscle enhancement. Okay, and again, each country has to decide what's important for them and pass regulations. Okay, that's where I want to stop.
Thank you, Professor Chandrasekharan. It's, I think you've captured it appropriately, where every country needs to decide where do they want to start, where do they want to draw the lines, especially in a country like India, where the disease burden is quite high. It's really critical that we start addressing our, the disease or the disease sector rather than creating super soldiers. But yes, going back to what Shamir said, well, it starts at doing good research, getting the most efficient genome editing done, reducing the off-target effects. All of this is going to contribute to a better tomorrow. Like with my experience, viral vectors have been in science for over a decade, or more than 20 years, I can say. But if you look at it, today they are seeing the light of the day for a simple reason that the technology around has evolved to tell us that they are safe and they are not as harmful as we thought it is. So it's the good part is genome editing has evolved and come to light at a time when there's a lot of allied tech which is really ready to address these questions that we are speaking about. Thanks a lot for your view about this.
Can I make one more?
Of course, please.
About the GMOs, okay? In USA, if you knock out a gene in a plant, that's fine. You don't need FDA approval. For those of you who don't know, FDA approval is pretty rigorous. It takes about 7 to 10 years to get it approved. Okay, so FDA has decided if you just make mutations by NHE, non-homologous end joining, where you delete a gene or knock out a gene, you don't need to get approval, and that approval can be gotten through your agriculture department. Okay, but the reason is because spontaneous mutations arise all the time. For instance, even in cows, this is horn locus mutations were there 500 to 1,000 years ago, and they have very small horns. So there are naturally occurring mutations. Of course, the talents have been used to make cows without horns, right? Now, once you introduce a gene, then that needs FDA approval, okay? So that's the US policy. I'm not sure how it's done here in India. Somebody else can fill that information. Thanks.
Thank you. And thanks a ton for touching on that particular point, because that will take us to Dr. Viva. Viva, your opinion, because you mentioned that agriculture is something that we need to really speak about. And I think Professor Chandrasekharan has just ignited the discussion about agriculture. Dr. Viva, over to you. Your opinions on this affair, please. Thank you.
Right. So before I respond, to what sir initiated the discussion in agriculture, I would like to just share a little bit of background, what has been happening in agriculture. You see, in agriculture, the way we have divided the three the products, the products of gene editing into three different categories. They are referred to as SDN1, SDN2, and SDN3. So the difference between the plants and the animals and the other, our medical applications is that in plants, we have the tools, to get rid of anything which is, not desired, things like off targets and other issues.
Yes.
Very serious. So those, we can do back crossing, we can do various tools available to get rid of all those off targets. So the thing is, the way it has been divided, the SDN1 and SDN2, the major concern while we think of the ethics or the regulatory issues is the presence of foreign gene. That has been one of the major issues in the whole paradigm of the genetically modified technology regulations. Oh, you have genes from different sources. So that is, even though they were deemed to be very safe, and a lot of safety studies were done, and I don't know, those proteins, they have been approved in so many countries coming from very, very safe sources, still there have been apprehensions. So in the gene editing, what we are seeing, a very, very useful point here is there is no foreign gene, and that advantage has been taken care by many countries. All the major countries like United States, the agriculturally dominant, countries like US, Australia, even Canada, or countries like Japan, many countries have taken a stand, and they have already modified their regulations to exclude the SDN1 category of plants, even many of them SDN2, exempted from their regulations. So, and this is because a lot of work has gone into it. There are many papers, and in the plants we found an advantage, as I said, that the precise nature of modification, if off targets, if any, there are tools available to get rid of that and get very, very clean insertion, deletions. And this has been a very useful thing. And we have several products, in fact, which have come to the market. So as I was sharing with all of you that we do a lot of activities and interact with the regulators globally. We had a very interesting webinar, a presentation by the inventors of tomato in Japan. And it was very proudly, they were displaying the gene-edited tomato, which has been approved by the Japanese government and in fact grown in the home nurseries. So even in India, so building on what is happening around the world, another very important factor which has come into the regulation and trade, particularly coming out of these ethical issues in agriculture, is that finally, when you have these genetically edited plants which have been modified using SDN1 and SDN2 category, the final products will be indistinguishable from the their traditional counterparts. So the issue comes is that if there is these are being traded across the world, one country regulates and the other country doesn't regulate, how are we going to actually distinguish them? There are no tools by which I can at least say find out through various advanced procedures that this deletion has happened. But whether it has happened through traditional breeding or gene editing or mutation breeding, that itself is a big challenge. So keeping all that in view, sir, in agriculture, the most countries, the stand which has been taken is that SDN1 category is actually being exempted by many. SDN2, I would say slightly lesser number, but people are moving towards that. And even in India, Shamir is also a member of that particular committee where we were all there and discussing about it. And initially in India, the regulations which came up, the guidelines which were released by the Department of Biotechnology, they were combined for plants, animals, and human somatic cells and the germline cells. But then the committee the Department of Biotechnology took a decision based on the comments by the stakeholder that plants, animals, other products have to be dealt with very, very differently. And the focus now is on having the regulatory guidelines for plants immediately, and then we move on to the other products. As our Dr. Sir was saying, that as the technology evolves, you have to observe and go with the advancements and develop the regulatory issues. Now, I was saying another thing which Shamir said was that the guidelines are actually made by a committee, or who is the one who is deciding on it, what is happening in the country. I would say I have been associated with the development of guidelines and dealing with these kind of ethical issues, even for GM, for several years within the government authorities. Also working with them, we look at everything. There are, properly documents are prepared, and that is important also. Because as I said, we go ahead and we say, no, we are going to regulate SDN1 type of products. It is the same product which is globally under research, and we are unnecessarily stopping the innovation in our country if we put those regulations which are not valid. And in other cases, if we are too lenient, then we might be, putting our populations to risk. So it has to be a very critical balance. And most of the times we have seen that there is a very critical evaluation of what's happening in the other countries. Harmonization of regulations is a very, very important area. And every country is, we have to ultimately, it is the commercialization and the trade issues which will come. So there we look at that, we look at the kind of research which is there in our country, and then only all these issues are dealt with in the policy arena. As of now, what the guidelines which are available, which are being developed and which are under discussion for plants, there is a strong thinking that SDN1 and SDN2 have to be exempted from the regulation, biosafety regulations, which are applicable to genetically engineered plants where there are foreign genes present. But in these particular cases, one has to ensure the applicant or the developer will have to will have to present the data confirming that there is no other insertion, and it is only it qualifies for these two particular categories of gene editing. So this is where we stand today. And I'll be happy to answer any further questions that you have.
Thank you, Dr. Viva. I have a bunch of questions which I'll come to as soon as we finish with Dr. Vishal Rao's opinion and understanding on this, because I just read about before the panel discussion a few things, and I have some questions. I'm just going to come to it. But Dr. Vishal Rao, over to you. We would like to hear about your opinion on genome editing, and your work and my work is quite close to each other, so I'm definitely looking forward. Thank you.
Thank you, Rohan. First of all, thank you for organizing this very interesting panel. At the first invitation, I was actually quite intrigued because I myself sit on two important ethics committees. One is with ICMR and the other is with our institute. I also play some role in the ethics committees with the ITBT. Now, this particular debate has not been a new one. It's a very old one. I think genome editing debate has been a very old debate, I think more than 2 decades. But what's interesting is the revisiting of this debate happened because of CRISPR, telling that it's a little more accurate and a little more easier. So I think there's a temptation to revisit this whole discussion. And I think for me, that is the way I looked at it, that I was told by somebody that it's going to be more accurate, it's going to be more easier. So I think let's relook at the whole thing. Now, I think Professor Srinivasan very beautifully alluded to the whole concept of germline versus somatic gene editing. I come from the somatic discussion. I'm not from the germline discussion. But yes, we do often have a few of our patients who are trained Google doctors who come and tell me that I have cancer, so can I prevent it for my next generation? But that's a separate germline discussion. Very, very few of them. Majority come for somatic discussions, somatic germline, somatic gene editing discussion. Now, if I look at it from a cancer perspective, I often say that there are 3 ways in which a gene has contributed to this discussion in oncology, either for diagnosis, prognosis, or therapeutics. Now you look at all the 3 areas, majority areas it has not helped. Okay, either in diagnosis, in prognosis, or therapeutics. So we've moved from genomics to proteomics, metabolomics to microbiomics. We are going into multiomics. But the truth is that In prognostication, yes, we've actually been able to kind of find out areas where we are getting some kind of signs, but not a complete conclusion. So I'll give you an example. P53 mutation, the guru of all mutations we've all talked about. And we did a small study here of 100 odd patients who had P53 mutations to find out that more than 2/3 of them were doing extremely well. In a traditional group of cancers, homogeneous, where almost all of them do very bad, 2/3 of them with that mutation did very well. So we realized that there's more to just a gene mutation than just a generic approach to saying there is just a gene mutation and saying that this gene mutation is a bad mutation. So we are not foolproof even in prognostication. Now take therapeutic target, like Professor Chandrasekharan said, if you spend hundreds and thousands of dollars, you get a nice comprehensive report which says, yes, this gene can be targeted by this drug, but how many of them will be able to afford that drug? That will be less than 1%. So therapeutically also you are in a minimized area, and diagnostically, I think CRISPR is promising a lot of potential for focusing some amount. Like I think the current discussion I've been doing with experts is on HPV utility for CRISPR. So having said that, I think we need to acknowledge the fact that while we need to keep discussing, researchers and bioethicists are going to keep looking at this area from various angles, and there is no final conclusion. But I think next couple of years we need to keep this discussion on, because we don't know when we may possibly make the breakthrough. It's like buying a property in Mars, because traditionally now Jeff Bezos, everybody's getting aggressive, so it's might as well that we sell this property in Mars and say be ready, because it might just click. So that is where we are right now. And I think I would like to close with 2 points. The slippery slope for us, I think, is in the area of the target effects, mosaicism, in the area of enhancements, and in the area of non-therapeutic usage. I think these are 4 slippery slopes I often feel are glaring at our face. So we need to be cautious about these 4 areas. And just to be, I think, to conclude, the 3 important regulatory areas where Government of India is now looking at very seriously, which is, I think, already in the role, is they've set up something for recombinant DNA Advisory Committee. They've set up something for Genetic Engineering Appraisal Committee, and something for Review Committee on Genetic Manipulation. So the coming days, I see that there's going to be a lot of attention given by Government of India for all these 3 areas. And we will have to closely watch how regulations are formed. And I think this group, particularly collectively, will have to have an important role to keep recommendations. Like I feel like the Nuffield Council or somebody has to keep guiding the government to tell them on where do we draw the ethical borderline. Thank you very much.
Thank you, Dr. Vishal. Absolutely agree that yeah, it started off like it's less complicated. It has less side effects, it has less target effects. But well, that's the glamour when it starts, and then when people understand, start doing deeper science, is when more information starts coming, and then there's a slight pushback. But I think that's the beauty of science, that it starts giving a lot of hope. The hope converts into more data, and the data eventually converts into either diagnostics or therapy or betterness of the products. So while I start a question, I encourage the participants in the conference to please post your questions in the chat, or the question answer session will be, the panelists will be happy to take it. So yes, so I have a question, Dr. Viva, and that's where I wanted to touch base, where if you look at, I believe that the 2 categories that you mentioned, of my understanding, this has been with the ministry or with the government for some time, and there seems to be a pause in the thought process, maybe. So can you throw some light on where, how many interactions do the government or the policymakers have with different think tanks, with different stakeholders, to understand what is the end user or what is the need of the hour, and accordingly act? Because if the meetings are going to be twice a year, it's slow. It's slow because science doesn't move by the year. Science nowadays moves by the days. So we'll have piled up information and then slow policies which are not going to match the requirements. So I have that question about how is the government approaching different think tanks on this? Thank you.
Yes, absolutely, Rohan. Dr. Rohan, this has been a problem, and particularly in the agriculture sector, also people are really facing a lot of, the challenges of science regulation not moving at the speed the way science is moving. So here in this particular case, the way as I'm aware of, I am not part of the earlier system of the Department of Biotechnology, but we all came in as part of one of the committee. Shamir was there and others were there who were to formulate particularly for the plants. So there has been some discussion going on for almost 2-3 years, which DBT had. And then finally they came up with a document, and of course this decision was taken that we will concentrate on the plants, and a document was prepared. As I understand, the developer ministries, like the Department of Science and Technology, Biotechnology, and Indian Council of Agriculture Research, they are very much, impatiently looking at, that the guidelines should be passed at the earliest possible. But I think the problem is somewhere in the Ministry of Environment. The Ministry of Department of Biotechnology had, made presentations in the Genetic Engineering Appraisal Committee. Those presentations were made sometimes in September 2020 or so. I think there is some internal discussions going on, but this is definitely a very, very major concern that the decisions have to come forward. Only then these innovations can see the light of the day. If the guidelines are stuck like that, and the last that we have heard is that the guidelines have been forwarded to the state governments for comments, which is again a big challenge. Who has the expertise and the knowledge within the state governments, and what mechanisms they have to adopt, and whether it has to go actually as per the regulations or not, that itself is a question mark. So this kind of problems are being witnessed. So I would urge here through these kind of interactions that our participants, our scientists and researchers have to actually, join hands and press upon the relevant authorities to bring out these guidelines at the earliest. I'm aware of several scientists who are sitting ready with their products, and they have to, move further, and there is no clear guidelines and uncertainty around what processes are to be adopted.
Thank you, Dr. Viva. I think a lot of this goes in circles where it goes from the scientist to the center to state. Yes. Then there's sort of a lag phase because of the lack of expertise. I think one comment to Shruti that when we organize such a conference where we talk about CRISPR-Cas9 and its tech, there might be the need to organize a dedicated meeting, a whole day meeting on ethics and keeping them individual, like agriculture, animal, veterinary sciences, human, and try to involve the stakeholders from state and central government, which will help them, understand or the pain point at the other end of spectrum. And I think it will be a very fruitful discussion. So, Shamir, what's your opinion on when I started off? Yes, I think Dr. Viva, you had something to say. I'm sorry. Okay, Shambhavi, over to you, if you don't mind.
Thanks. So I have been part of two very different interactions with DBT over guidelines. One that Dr. Viva just spoke about, which is the risk assessment for plants, which seems to have got into this bureaucratic kind of stoppages over the past year, more than a year now. The other is for the gene therapy product guidelines, which are for the human gene therapies. I started working on this in 2017. I think DBT ICMR had their first meeting also a little earlier in 2017 or in 2016, which was on the same lines that you just suggested, Rohan, where there was an entire day of understanding about the technology, the ethics, etc. I wasn't part of that. Manisha was, so I wish she was here, and I think she could have shed more light on this. And then again, it kind of disappeared off the radar. We did not hear of what was happening with the guidelines till they came out in December 2019. That was a remarkably different experience because so all of these guidelines sometimes are shared for public consultations as well. And the gene therapy ones, which are so important for India, were out for consultation for, I think, 5 working days. There is no clear-cut mechanism by which you can also follow them, right? So they come up on the DBT website, which means you have to go and check if you are, and anyone can, and all of us, right, can comment on these. So we did write in a consultation for those. We never heard back, and I think a month later, the guidelines came out exactly as they were, as the draft was. So I really doubt what kind of public engagement happened for those particular set of guidelines. The risk assessment for plants was very different. There was a meeting in Delhi. I'm sure there must have been a meeting in at DBT even for the other ones. But that seemed a bit more comprehensive. They showed us all the stakeholder comments, etc., which was very nicely done. I think that particular part of public engagement is something we have to bring in for gene editing, particularly from an ethical point of view. Thanks, Varun.
Thanks, Shambhavi. What I've observed is when it comes to human application, the government and the policymakers are obviously more proactive because, it touches factors of rare diseases, genetic diseases, life-threatening diseases like cancers. And there's a lot more activity happening when it comes to human application. There's also much more control, which is good, which is great. Around 4 to 5 years back, I'd held a workshop at KIIT Bhubaneswar on genome editing, a hands-on workshop for a whole week. And I was surprised to see, I'm a person who's never worked in plant biotech or even, I've not taken botany, I'll put it as simple as that. And what I realized is majority of the participants were actually people from the agri field because they were so keen on using this tech. I mean, as Dr. Viva said, there are people sitting with products in their hands, which have really bettered the field. And unfortunately, it just, everybody's waiting. And I think it's really time that something has to be done on that. Now, I would like to take some questions, which I can see in the chat box. One, Neil Sharma has to say that, according to him, what has been happening is we have, because of the lifestyle, there's a lot of change and diseases being introduced into our lifestyle. And the question goes about, will there be ever genome editing intervention, through embryos and IVF, and is that the way we are looking at in the future? I personally say not so soon, but I would like to hear from panelists because we've already spoken about this, where we have to first cure the diseases at hand and then we go a step ahead. But comments from panelists, please. Where do we go from? How far? I'll put it this way to the panelists. When we are looking at embryo-based genome editing, is there an approximate figure that any one of you have in your thought out of your experience, just out of curiosity? Five years? Have I lost connection? No, I'm here.
In US, the policies are clear-cut. No germline editing. Okay, so I don't think lifestyle changes, any genes affecting those, they will not allow it. Okay. Like I said, if the technology improves a lot and it's very safe, it probably will be done mainly for diseases, so it doesn't affect the future generation. I doubt it very much that it will be allowed for cosmetic changes or eugenics, as we call them. So I agree with that.
Yep, and I still strongly believe in the concept of survival of the fittest. That's one way where nature keeps a control on how we exploit nature. Comments about gene drives. I know that there have been studies going on with genetically engineered mosquitoes. Any idea about, that it would be great to understand. And if I'm not wrong, there was a study in somewhere in Maharashtra by a company. So any comments about gene drives, how that can help us, India particularly?
Gene drives, the idea is very simple. You introduce the CRISPR-Cas9. So when it mates, it will pass on the gene. The CRISPR-Cas9 will be passed on. And when they have progeny, it will also carry the gene forward, and it will propagate through the whole population. And it will cut up the gene so it cannot produce any more, either no more female flies, something. So I think they released it in Florida, in US. They did, but there was a problem. Some of them developed mutations, right? The CRISPR, the specificity of CRISPR chain. So you end up with a mutation. So you end up with mutant mosquitoes. So they're trying to improve it. That's how science works. You run into a problem, you try to modify and develop further. So I think it's at very early stages right now.
Yeah, absolutely. And I think this is the kind of worry that puts a lot of, common man, the layman, in fear that what if something goes out of our hands and now we can't control? I mean, as our last speaker had mentioned, you can do a recall of a defective car, but you can't do a recall of these mosquitoes and mice and rats and animals which are out there, and you can take them back. So that's something which is of real good point. The next point that somebody has raised that about DIY CRISPRs. Now I know that in the US you actually get DIY kits where people try to do some experiments at home in their laboratories in the garage. Where does India stand on it? And to my knowledge, it's not something that is very popular in India. It's not. It is controlled. Research of this sorts is done in research institutes, colleges, where there is certain level of expertise to look at what is happening, and that's really important. But comments from the panelists to understand, is there a way where we think that such things should be introduced in India, where you can buy a kit of Amazon and try to do some level editing in bacteria and see GFP on a plate? Do you think we as Indians are ready for DIY kits at home, just to learn?
Rohan, I was reading about this, and quite interestingly about from some of my friends' kids from US who wanted to change the color of the bread when they eat their sandwiches and things like that. Quite interesting. I'm kind of more, I'm now talking more from an interest point of view. I would love to look at ordering it on Amazon. I've been doing a lot of Amazon shopping, so I don't mind trying it out and seeing what happens. But I think somewhere we need to know that each vendor who sells this kind of a thing has certain amount of liability on them. More than anything else, it's more than I think the seller has a liability here, and US is the best known for understanding liabilities. When they sell something, they know how important liabilities are. So I think if US has understood it well, and if they've kind of done a deep dive into it, it's mightable that we relook into this and see where do we draw the line. Because I think where I see it is that some of these things are going to come. Like, for example, I'll just classically give you an example, robotic surgery for us. 5 years back, I remember my chairman telling me it's very important, try it out, look at it. And I said, no, I'm very happy with my results. I don't want robotics at all. He convinced me for a couple of months saying, no, you should look at it. And I was saying, I'm very happy with my old technique, same results. But one fine day I had to go to Korea. I got trained in it. Now I'm doing robotics so frequently, and I realized that, yes, there are certain advantages. But the mental block that we sometimes have to embrace a new technology comes possibly from education telling us this is what you've been taught, follow it this way. a time for us to relook into some of these new things to see where we have to draw the ethical lines. And I'm saying people like us all who will be sitting in ethics committees, participating, taking guidelines, if we have no experience on something, then to comment on something on that becomes more difficult. That's where I would stop. Thank you.
I absolutely agree. And I feel that's the curiosity that such DIY kits can enable, enhance, and promote people to get into science is something which is fantastic. It also is there's always a fun factor. I mean, I believe everybody knows about the fold scope, which is a small, folding microscope which you can literally unfold out of a compost box. The old school ones, I think it was $10 or something even lesser. But it really fueled a lot of imagination and science. And but when you talk about the seller and liabilities, right? That's something that it's really important out here because, well, if you see our Indian roads and the vehicles with crazy amount of loud horns and flashlights and everything, seller in India doesn't really have any liability. Once sold, the person disappears. And I think that's where regulations again come in picture, where maybe there is some route through which these things can be introduced in schools way early, where people get excited. And the science fairs that happen in school are places where I think this can start off and then eventually go home. So definitely agree with the part where it can, I think, really help stir up a lot of thought process. Great.
So, just to add one, I think liability in India is increasing and monitoring is also increasing. From routine monitoring to Pegasus to everything is increasing. So I think we are all on the guard right now. So hopefully the coming days will be even stronger.
One thing we can observe all the while is that our, as you say, awareness is increasing. There is always a fear of being, fished. So yes, and I think it's there's a lot of effort from the government to tell us that you are at risk, be careful, and that is something which is really valuable that is happening. So yes, questions from Karthik about DIY biohackers. I think it'll happen. Give it some time, it'll come, but with conditions apply, definitely. Great. Shruti, you're saying that we can take questions from audience. How can we do that?
So there's Dr. Devashish Rath has raised his hand, so he can actually unmute himself and ask the question.
Perfect.
Yeah, thanks, Shruti. So I was I have been listening to this talk that there are guidelines and regulatory framework that is being put in place. So I would like to know from some of you, maybe this was discussed and I joined in late, that is there any real information on when can we expect these guidelines, particularly with related to plants? Is like, at what stage it is, or some of you might be involved in that. Unfortunately, when I talk to a lot of CRISPR biologists who have been doing CRISPR for a long time, none of them have been involved in that policy making exercise. So I guess some of the clinicians or some of the people who have dabbled with gene editing in a conventional way before probably would be involved in this exercise. So is any information on that?
Shall I?
Yes, please. Yes.
The guidelines for plants, I can at least tell you a little information. See, we have a system in our country. There is something called rules for, which were drafted way back in 1989, rules for manufacture, storage, use of genetically modified organisms and hazardous microorganisms. Genetically modified organisms and hazardous organisms. Now, what has happened is that although the Department of Biotechnology and others were very keen to have these guidelines put in place at the earliest, but the scope of these rules, although they were made way back in 1989, and they were expected to cover they were actually made for hazardous microorganisms. under the Environmental Protection Act. But around that time, 1989, the research in genetically engineered organisms was starting in India. So this two were clubbed. The genetically engineered organisms were included in these rules. But if we look at the scope of that, there is just one additional line written there, that any other new technologies developed over the course of time. So that is where the guidelines have been stuck, because you have to seek an exemption from these rules 1989 for an alternate mechanism. And this is under the consideration of the Ministry of Environment. There will be a little bit of the legal process. There are a few committees which look at these guidelines before they are approved. 2 or 3 important levels have been crossed, but it is, with the genetically engineered appraisal committee, which had to take a decision followed by the legal process. We understand from our information that everybody is very keen to have these guidelines at the earliest possible. Of course, no clear timelines can be set, but I can only assure that, this is my feeling. I can't assure anything, but my feeling is that we should have them, let's say, in another 6 months or so in place.
I will be a little more specific, like particularly to give you an example recently with a tomato example coming from the Japan. So
Yes.
like what Indian researchers and particularly plant gene editing people are looking at with bated breath, I should say, is that whether it will be like in some countries they have very defined that if it can be done with conventional technology, anything that can be achieved with conventional technology, and if CRISPR just accelerates it, it is fine, they will allow it. It doesn't go through that complete ban or something. But anything that, but there are some other countries, for example, Europe, which have taken a very clear stand that, okay, anything to do with CRISPR will fall under GMO. So is there any clarity on that?
Yes. So if you look at the, minutes of the meeting of the GSC, which have come up, where the guidelines have been discussed, it has been clearly indicated that what has been recommended is that SDN1, something like Japan, or even more than Japan, because it has been indicated that SDN1 and SDN2 both should not be treated in the same way as genetically modified crops, will not be treated. The ones which have the SDN3, which has the foreign genes, will be treated as GM crops. That is the thinking as of now. But till the time we get the final regulation, if some change happens, we do not know. But I will also like to share with you what is happening in Europe. Recently, I was, part of a discussion which was initiated by a European agency, and where in fact, Dr. Jennifer Doudna also spoke, and she prompted people to, use this technology effectively for plants. So there, we heard a lot from the Europeans, including the UK. Both UK and EU are slowly moving towards exemption of these SDN1 from the regulation of the GM crops. UK agencies, DEFRA, which is the agency there, Department of Environment, they clearly said that maybe before they said very shortly, within few months, you will have their regulation. They have already completed their consultations. In Europe, European Food Safety Agency has already given their opinion. European Commission has come out with a report where they have said that except SDN3, which should be treated as GM crops, the other two do not pose any risk. So we are really hoping that even in the Europe, unlike the GM, there has been a kind of a clear-cut 2 different groups. We are really hoping that there will be a turnaround shortly in Europe also. So let's hope for the best, that we have a clear-cut regulation. Thank you. I hope I answered your queries.
Yeah, thanks a lot.
Can I make a comment on about doing gene editing at home using bacteria? You don't need genome editing to do bacterial transformation. They take up DNA pretty well, right? Only in some organisms which don't take up, maybe you need it. So I wouldn't worry too much about using genome editing for transforming bacteria. Second, even if you have a kit to do it at home, there are a lot of steps involved. It's not as simple as it's made to sound. You have to, for CRISPR, you have to have CRISPR, then you have to have sgRNA, then you have to, and to transform plants, it's very difficult. I'm sure many of you know that. So it's not that easy to do it at home.
It's not the 2-minute Maggie that we make.
You're right.
It's a biryani which has to be cooked over a long period of time with absolutely appropriate ingredients. Absolutely.
What I wanted to say was you need some expertise to do it, and you will need some money also. So it's right. You have to buy a lot of things.
But saying that, Professor Chandrasekharan, I can definitely assure you that there is a sizable population which will not, that's something which may not hinder them, because buying instruments off the shelf is quite easy now. So if with an appropriate guidance, it's actually not a challenge, is what I've realized. You have people who are really passionate about DIY who would like to put such things in place. But yeah, it has to. It's not easy, no doubt about that. No doubt about that. Thank you. Thank you for your advice.
But I'm of the opinion it should be taught in high schools how to do these experiments.
Yep, absolutely. Yeah.
To stimulate their interest.
It starts there, and then it grows up to become great science. Dr. Bindu, you have a question. You've raised your hand. Please, you can ask the question by unmuting yourself.
Thank you. Madam, I have a question. Viva ma'am, I have a question for you. From 2006, we have seen in the IGMORIS website, IGMORIS website, that from 2006 onwards, we have been doing field trials for GM crops, various crops like potato, cauliflower, and many more. And as of now, only BT cotton is being approved. So where do the products escape? What is being done with those, the products? Because we know that for certain crops, the harvest time is very less. So what do they do with these the products of the GM crops?
Yeah. So, unfortunately, there has been, as some of you may be aware, but I'll just repeat, after the approval of BT cotton, there was a great success of it. Now in our country, more than 90%, 94% actually, area is under BT cotton. So after that great success, there was a lot of enthusiasm among the agriculture scientists and also the supporting agencies, ICR, DBT, they supported a lot of projects. And as you said, there were several crops which were developed in the lab. And I would be saying, you are saying 2006, even in 2014, we did one survey, with the under one Ministry of Environment project. And there were actually almost 85 different plants, which were under some, research or some activity within India, 85 different plants, including medicinal plants, herbal plants, and so on, where there was interest for genetic modification. But unfortunately, what happened in 2009 when BT Brinjal came, the then environmental minister, Mr. Jairam Ramesh, he put a moratorium on it and on that particular event. And subsequently, there was, and one of the reasons of that was like a polarized debate in the country. The Greenpeace was very active and they had really activists put a lot of pressure, and the government should have pushed back themselves at that time. Subsequently, we were really looking for a breakthrough again, when Professor Deepak Paintal in the University of Delhi, he came up with a mustard, genetically engineered mustard, which has been, the final dossier has been prepared through a public sector institution. There was whole rigorous regulatory exercise done. I'm really fortunate to be part of that. And everything was going very well. And it was hoped that this time, because all this multinational, questions and all those are not there, this is our own product. It is our own patent. Dr. Paintal has a patent on that. The country needs mustard very badly because we are importing next to, the petrol and the gold, the next import bill is the oils that are being imported in large quantity. And here is one technology which can actually give you 25% to 30% higher yield. But somehow, there has been again a lot of public debate, and several activist organizations, some of them are very, very, I would say, they're very active and they put a lot of things in the public press. And we were, in spite of that, it seemed that the government is moving forward. And then the then environment minister, from the ruling party, he actually died. And then, of course, there has been no decision after that. So there are all kinds of questions being put up. Things like, the bees, the beekeepers had a question on the honey. And, many of these questions have long been answered in the countries which are growing. The product similar to what Dr. Painter has developed is already there in the market since last 25 years. So somehow, this GM technology has been part of this polarized debate, and that has done a lot of damage. Many of the products which you have mentioned have either been shelved off or they are sitting in, some scientists' labs. They are just maintaining them, waiting for a conducive policy environment to come where they can, take it forward. For the field trials, a requirement was introduced by Mr. Jairam Ramesh to take permission from states. That has become another stumbling block, You have to run from state to do the trials, which people are not able to do. I know Rohan is looking at me, I'm taking too much time. But so this has been definitely a little bit of a negative thing for a very promising technology. And we really hope at some point there will be a positive change, and all these technologies will see the light of the day. And we really need them. There are, like I gave you the example of mustard, there are many others. Our chickpea and pigeon pea, dal that we eat, they are subjected to a lot of damage. And then the challenge of climate change, now you see so much of climate change effect is there, and you need drought tolerant, salt tolerant, and whatnot. And that cannot be done without the use of genetic engineering or gene editing or any such innovative solutions. So we look forward to a positive policy environment so that it can move forward.
Okay.
Thank you, Dr. Viva. And I can absolutely see the, desperation of sorts when it comes to the requirement, and we really, I think, need to work with it. I hope, Dr. Bindu, your question has been answered.
Yes, one more question in continuation to that only. We come across in media that clandestinely or in many parts of India, the GM which is not been approved is being cultivated, like GM brinjal, and there were many other crops as well. How far is it true, ma'am?
It is true, because you see, particularly in case of the herbicide tolerant cotton, almost, they are saying 30 to 40% area is under herbicide tolerant cotton. Because, if there is our borders are very porous. BT brinjal was not approved in India, but it is approved in Bangladesh. And Bangladesh farmers are growing, it is being sold in the market, seeds are freely available. So how do we expect that people in West Bengal who are just across the border will not like to use a new technology? So these are the kind of things which are happening. Herbicide tolerant cotton is very, very successfully used around the world. And here we have so much of labor problem. Agriculture is facing a lot of labor problem. The use of herbicide tolerant crops can really help. So the farmers have just gone ahead and are using it illegally. But the problem there is, the technology developers don't get any incentive. Then there are always, when such technologies come, there are package of practices developed by the research institutions, universities, for every location. But here what you see is when it is an illegal market, you miss out on all those instructions and a proper use of any product. So they're actually, damaging our agroclimatic system by this misuse. But everyone knows that the issues of farmers are very sensitive, and nobody, takes any action. And this is the situation that herbicide tolerant cotton is almost 30 to 40% area under illegal cultivation. So it should be an eye opener, and there should be a positive environment, but we'll have to wait.
Thank you, ma'am.
I think this particular event was also an eye opener, and such events are the one which sort of put government in the fix where they start pushing back when illegal activities happen. But well, as you say, our borders are porous. There's not much that is under our control. Great. So moving over, Mr. Neil has a question which I seem to have not quoted properly. So sincere apologies for that, Neil. Shruti, if you can unmute Neil and he can pose the question to the panel.
Yeah, he's allowed to talk. So Neil,
Yes.
Please go ahead.
Neil, please go ahead.
Can you hear me?
We can hear me. Yeah, you can.
It's not a question. It's more of a thought I had that, before the advent of modern medicine, people who were genetically fit were able to get married, and even the children they had, even those children that survived were the children that were perfectly fit. Now, with the modern medicine, genetic disease or disease itself is not a foundation or a hindrance to marriage. As a result, most of the genetic defects is accumulating in our population. So as an evolutionary point of view, I believe that our genes are becoming weaker rather than becoming stronger because of modern medicine. And I personally believe that when this technology becomes good enough, I believe that IVF and embryo editing will be the way of conception, and natural conception probably become obsolete when this technology advances. It is my opinion, and what would be your opinion in terms of? I hope you got what I said.
Thank you, Neil. If any of the panelists would like to answer the question, basically, if I get it right, you're trying to say that we are progressing towards genome editing and artificial way of conception because of our evolutionary trajectory in terms of genome, the way our genomes are evolving.
Yeah, I mean to say that, let's say, someone had a diabetes 200 years ago, that person will not survive. That person would be dead by the age of 5 years. But today you can survive when you have kids. So, you may marry someone girl who has some other disease. Now, these genetic defects are then accumulating. So basically the genetic defects in the population is basically growing. So, we'll have so much of genetic defects and so many of these chronic diseases that ultimately, embryo editing will become the norm. Governments would have to do it to, relieve the burden of these non-communicable diseases from the population. That is what I meant to say. Today, if you have cancer, as a childhood, you can get married, but probably, 200 years ago, you would be dead. So basically, our genes are becoming weaker. Our evolutionary, it's not that the fittest is surviving. Everybody is surviving. It's not a wrong thing. It's a good thing. But that is making the genes weaker in the overall population. That is what I mean to say. So it will become the norm, and governments would have to do it.
Let's hear from the panel what they have to say. Comment. Vishal, I would like to
Just
to
start, I think it's a very thought-provoking question, and I think it's brought out this discussion which has been latent in many of our minds, correct? Because we are filled in an environment of carcinogens today, or pollutants, adulterants, chemicals. So everything is seeming like it's doing environmental food. Everything is editing our gene in some way today. Now at this juncture, today, if you look at the current consensus, the debates are currently around saying that the current level of technology seems to be not superior to IVF or any other kind of neonatal screening at this juncture. Should we aspire towards that time when germline, and that's the whole ethical debate about. Researchers, bioethicists are all talking about that particular, like they call CRISPR nature's gift that has been done to possibly do it. Have we been able to do it? And the same thing happened, and I must tell you all, when from Watson and Crick's time till when Craig Venter talked about the whole genome sequence, he said, give me the thing and I'll tell you all the 23,000 genes and what they do. At the end of it, they said, no, I think it's about epigenetics. And at the end of epigenetics, we said it's microbiome that is actually the target. So we are on a moving target. We've not decoded it. And we finally found that mine could be an epigenome. So this is going to be a moving target, and that's one aspect. And I think the other controversial aspect was marriage. So I think there are two separate things he has to segregate from this. But I agree that that in the coming years, these are going to be continuing debates that are going to happen. But keep discussing this, and I think this debate will keep going on for the next 5-10 years, ongoing.
Great, thanks a lot, Vishal. Perfectly put about this. Shruti has a question. Shruti, over to you.
Sure. I actually have, I typed both of my questions in the box, but I thought I will ask them since Dr. Vishal is already talking. I will ask him the question first. What are your, have you ever discussed with patients how receptive they would be to such therapies, to Cas-based therapies, or any kind of genome editing therapy if they want to fix, say they know they have a certain mutation and they're highly likely to get a certain cancer. So would they be receptive to such a therapy? So that would be my first question to Dr. Vishal, and then I have a question for Dr. Shambhavi as well.
So I actually saw that question. I thought it was a difficult one. That's why I didn't want to answer. I hope that
okay.
Yeah, so see, I think the way I would look at it is today, people are asking for CRISPR and this kind of a thing because of Quora, Google, and other things, and they've come to know of it, not because it's available and some center is actively marketing it currently.
Right.
Come to know it by reading, because journals, articles seem to keep talking about it, and newspaper extracts kind of give this hope. So they keep coming to us to ask. So for example, you've asked about BRCA. Most of the discussion around BRCA has been, does this give me a little bit of more certainty than just a generic BRCA thing? Because there is a lot of variants of unknown significance, which has been promised by CRISPR to say that it gives a more accurate prediction for women. So does it improve certainty? These are the kind of things that have been asked to us. So most of the times, when we talk about genomics, and this has been my personal experience with most patients who ask a lot of these questions, if you tell them the cost about it, the questions slowly reduce. They don't ask much of those questions, because it's the cost that becomes the innovation there, and the utility of it. Like many of the times in oncology, I tell, okay, there is this panel which you can do 100 gene, and then you analyze this, and then they say, okay, sir, and after that what? I said, nothing much. I just give you a little bit of the Janam Patri in detail, and that's it. I can't change much of your destiny. So then they kind of back off. So that's where we are. But I think our continued hope is that I think if we are able to stand in the shoulders of these giants and make some progress to targeting, and that I'm already seeing at this time, I'm already witnessing it, that while I laughed at it 10 years back, today I'm seeing some targets are available, and that patient whom I told you have 1 month to live comes and meets me after 5 years. So that's a reality today. So I'm not being pessimistic about it, because I think we need to embrace this area with a much more openness. And what has not been dealt with most strongly till now is what's on Rohan's background, the immuno aspect of it, which is very, very relevant. And I think that is also something that people are working around with CRISPR, in terms of CRISPR-based immunotherapies, CRISPR-based discussions on how to understand the interplay between genomics and the immunology is very, very interesting at this juncture, which needs to also be dwelled into through organoids, through other modelings that we can understand.
Okay, great.
If I can add that to what Dr. Vishal said, like I am a pathologist by background, and genetics is coming in a big way. Like CNS tumors now, morphology is obsolete now. It's basically all your CNS, all your genetic mutations, and basically pre-implantation diagnosis is the next big thing that is coming up, and you can have all those inherited disorders. But right now it's more related to single gene disorders, and nobody knows how to do it for multiple gene disorders. So that is a thing. I just wanted to add, because I'm a pathologist by background.
Sure. Okay. So I had a question for Shambhavi, partly because I know she's very passionate about gene drives. And can you share your thoughts as well about gene drives, and how in a country like India, maybe, right? What would you see foresee the future of certain gene drives, which are, say, CRISPR-Cas based?
Thanks, Shruti. Yeah, so I've been following this gene drive thing since 2018. Like Rohan mentioned, I had no idea about these trials in Jalna. that have been happening for over a couple of years. And it really had to dig deep on the Oxitec website to actually find where the field trial was happening, how approvals had been got. I still don't know who approved of these trials, etc. And then obviously we had some more research happening around in Bangalore as well around gene drives. My take on gene drives is that there is no market recall, right? So if they're out, how do we build in liability? Something that we have spoken throughout this panel. Whose liabilities if something goes wrong? Second is, do we have the capacity to monitor as a state if something goes wrong, when something goes wrong? If we don't have enough efficient data systems to, at this point of time, tell us how many dengue cases there are, how do we create a mosquito that is going to stop dengue if we don't know where the hotspots are? So how do we build that capacity was my first question. And then finally, as a consumer, if I say that I don't want to eat a genetically modified organism, I have the choice of not buying, right? There is regulation that says that you have to write on the label of a food product that this is a GMO or not GMO, etc. As a citizen, if tomorrow BBMP decides that we're going to trial genetically modified mosquitoes here, what is my choice? Apart from leaving if I do not agree to this, right? So public engagement for this particular application, I think, is extremely, extremely important. We cannot club this into any other application for genetically modified organism because it just is going to have so many other unintended consequences that are related to it. And I don't think we have done enough to kind of raise awareness about this point at all. So liability, I think, is going to be most crucial. Who takes liability for genetically edited mosquitoes? And again, this is a very interesting area because we have other solutions. It's not like we don't have. Sri Lanka, for example, smaller country than us, I agree, but has been routinely eradicating mosquito-related diseases. China eradicated malaria recently. Uzbekistan has eradicated diseases which are mosquito-based. So there are other countries, landlocked countries of bigger mass, which have done it. So if there are other potential solutions available, how do they rank up in terms of something whose unintended consequences we might not be able to tell? So I think that kind of policy research is still required.
Can I?
Yes, Dr.
Yeah.
Yes. Actually, gene drives is a very dear subject to me also. So I thought I will just add a little bit of information. I'm part of the gene drive outreach network, which is working very extensively, mainly in Africa. So the thing is that, like Dr. Vishal said, very rightly, that technology is moving very fast. So something which we think today is there, and these are the problems, the other day solutions are also there. So similarly, we really need to be watchful of the technology developments also which are happening in the field of gene drives. There has been, of late, further research done on it, on basically what you said, the recall product. This one, even gene drives are for population suppression and population elimination. So for population suppression, we are able to even have the drive that will multiply only for 10 generations. So that after that you have to introduce more, or they have just gone away. Secondly, you can introduce another drive to, sort of recall your earlier drive. So there are a lot of advanced technology options which are becoming every day. And I think it is going to be very, very useful technology in future. So while there is no debate on the public engagement part, it should definitely be there for a technology like gene drives. But also, we have to be very, very careful about the information that is available in the background. The Oxitec technology is not gene drives. It is genetically engineered technology. And there you have to introduce, from time to time. And if there is no tetracycline in the atmosphere, they will just go away. So there are ways to recall, very, very effective ways to recall in that. And this technology has been very successfully used in Brazil. And the third point, things about, eliminating this one, there are definitely lots of tools, and gene drives will be just another tool. Nobody is claiming that gene drives will replace all other tools available. We need to have a lot of tools in our toolbox because to use them as and when it is required. So even countries like Sri Lanka are very, very actively working on Aedes aegypti. They have come up with very good technologies using genetic engineering and gene editing. And in fact, we are very surprised at their developments. I have prepared the guidelines for their government on how they are going to use it. And shortly they may be actually conducting the field trials. So the thing is that, we cannot be away from these technologies. They are coming, they are knocking at the door. So the best thing is what you rightly said, do the public engagement, think about their judicious use, safe use for your country, but we have to continue innovating and keep a watch on what is happening globally because things are really moving very fast. Thank you.
Thank you. Thank you, Dr. I think I really like the way Dr. Viva put it. One tool is not going to solve any of our worries. All that we've discussed today on the panel in context of diagnostics, therapeutics, agriculture, everything, absolutely. It's multiple things that come together and make a rock solid tech to eventually translate out and be rolled out to the common man in terms of a therapy or a crop to be eaten in multiple ways. We are actually over time, but there are two questions which are the last set of questions I'll take from Dr. Sanjay Ghosh, where he says, while we have certain policies already in place for gene therapy, do we need finer policies on genome editing? Is one question that he has. And then the follow-up that he says is that without a very crystal clear policies, is it good to conduct research, without these policies in place or these partial policies in place? It will be great to hear from the panel about this. I strongly believe that it's important that we need finer policies. And if you look at the policies, they were actually starting to filter out and become very specific. And I can speak from my experience at Immunil that how policies have gone from cell and gene therapy to CAR-T therapy, and it's an evolving process. Dr. Vishal, I think you had comments, and I would like to hear about it. Thank you.
At the beginning, I said about this thanks, Dr. Sanjay, for bringing it up. I had earlier said that there are already one advisory committee and there is 3 regulatory committees. Now, the 2 regulatory committees which are there, one is on genetic manipulation, another is on genetic editing. Both are at the center level, at the federal level, and there is one at the institute level, which is the biosafety committee that they've set up. Now, I'm a part of one of these, so I understand that we have responsibilities, but there are less guidelines. Now, I think the onus here also lies on us at this juncture, maybe, and this is a cue. I would request the organizers of this particular conference to say if you can come up and sum up this entire thing and put it up into a document. I'm sure as a member secretary of ethics committee with ICMR, I'll propose it. I'll put it forward and front end the thing, or have all the people come together and we can use this as an opportunity of the consensus from this discussion. If somebody could put it together and present it to the government, to these committees, and say, we did a national conference, this is what we believe should be done, and this is some of our recommendations. I'm sure this is the right time for us to push it forward as a part of an action plan.
Great. Thanks a lot, Dr. Vishal. Any comments about whether we should do research without CRISPR policies in place? Well, I feel that research is research. Science should not stop. Yes, it should be controlled, and an activity like the IBSC committees, which are set up by every institution, really help keep a tab on what is happening. Okay, and so there's a record of every activity that a company or a research institute do with the respective with DBD when you conduct such activities. So yes, research should not stop, according to me. That is what is eventually going to drive things. Right. Dr. Sanjay Ghosh, actually, you may unmute yourself and say something.
Yeah, so I just wanted to say not to the research. The research should go on, I do believe. My thing was that translational products and translation research, because there comes the guidelines, right? I mean, if we have to trade using CRISPR, technology, anything that we develop, should we do and put money? And I know government does support that kind of research, but to me, it is kind of counterintuitive that the government doesn't have a clear guideline, but is telling or is expecting translation products to be out of it, and we don't know what will happen to it.
I think the point Rohani made is a very valid one. A lot of the time when we face this, I often go back to international guidelines, because at least if there is a precedence of some sort, then that is our respite at that juncture. And I agree that there are a lot of lacunae at various areas which we cannot, these lacunae cannot be addressed by us. But at least if there is an international framework, then that's a good starting point for us to address it. That's what I would like to say. That if we at least have an international framework, we refer to that and continue moving forward, because then we have a reference guidelines when the government, regulatory, or any approving body asks us a question relevant to it. I hope that answers it.
Yes. Thank you, Dr. Vishal.
Last question from Dr. Viva. We are just 3 minutes away from
this is for Dr. Viva Ahuja particularly. So extending what Dr. Sanjay Ghosh was telling. So I'll just give an example that some years back, I just asked one of the companies who are providing Cas9 and gene editing services. I said, okay, I have a bacterium and I make a change using the CRISPR, and can I sell it in the market, and do I own the IP? So they said, no, you cannot, because the moment you use CRISPR, the IP rests with the company. So I then said that's no use, because then I will use a conventional technology. We have recombination systems and everything available. So while Indian government is promoting a lot of this gene editing research, this is one area where I feel we are heavily restricted by the overarching patents that have been granted to some of these companies. So maybe Dr. Viva and Dr. Rohan can together answer this. So what do a small time Indian researcher who wants to use this technology and is able to find a jackpot because he has metabolically engineered a strain or something, so what does he do about it?
This is definitely a challenge. I mean, there are a lot of discussions going on, how we can maybe as a country take the IP. There are some discussions we heard in our forums, but definitely this is one of the very big problem. You have to go to the patent holders and take the license, and they give the license for research, but then it comes to commercialization. There are different terms that you have to negotiate. In fact, we heard a very nice example I would just like to share here from our Japanese inventors of tomato, If we can, I can even share one-to-one their slides, and other information that they shared. So when they were, of course, you are talking of a small time company, but this is from the University of Tsukuba. So University of Tsukuba had to create a small company, and they have negotiated with the inventors and taken the patent rights before they could actually commercialize this gene edited tomato that we are all hearing about. And Dr. Izura, who is the inventor, he shared his experience about it. Of course, he did not share the dealing, that how much money they would have paid or whatever they would have done. But we can definitely take guidance from them. This is definitely an issue Dr. Devashish, you have rightly raised. And there has to be some more discussion on this, on how to help our startups, our inventors, and who are keen to translate these technologies. You see, just funding the research will not end into the products for the society. So this is definitely a burning issue.
Thank you, Dr. Viva. A quick comment, Dr. Devashish, that when you look at IP which can be generated in your lab, it may be an academic lab or a small company / startup. The value comes when you can actually find a buyer, as simple as that. And that would really help if you wish to transfer it out else there are investors waiting to pump in funds. And the recent move at IIT Bombay, where Dr. Rahul Purwar had the first CAR T cell therapy administered to a patient at Actric in TMC. His startup got funded by a larger biotech pharma company, took a stake in his startup, and has given a route for him to commercialize the therapy which was developed in his lab in IIT Bombay. So that's the way things can move ahead. As with respect to the CRISPR-Cas9 patents, I think it is still quite difficult to pinpoint who owns it and how you can license it in. We quite some time back had spoken to, I think it was, yeah, we had spoken to MIT and we'd gotten numbers which obviously were not something that we could digest. So, but yeah, that and again, how have the patents been protected in India is something that you need to understand to figure out whether how you can eventually use them to commercialize a product. Great. It's 6:17. I'm going to put a pause to this panel because unfortunately we have to, because it's well past sunset, and it's a great, it was one of the finest discussions that I've had in a long time. I have to thank each and every panelist and participant of this meeting. Shruti, my job here is done. It's over to you now. Thank you.
Yeah, thank you. I feel like I could keep talking about this for the next hours. I've loved the discussion, and I've been getting messages on my WhatsApp, and everyone's loving it. But we have to end. We've been listening to science since 9 this morning, so we I think people are also tiring out, and I'm sure I don't want to hold you up. I'm sure you all have other engagements, but I really think very, very relevant points were raised, and I thank you, Rohan, a lot for moderating the discussion, and I really thank all of the panelists for taking their time to be here and discuss their viewpoints. I think we've had some very interesting discussions here, and Dr. Vishal, I think what you raised about how this type of a gathering of scientists, if we can actually move this forward also. So we will do this. I think we will go rewatch the video recording of this discussion, make all of the relevant points, and maybe we'll come back to you as well, all of the panelists, to get your input. And I think it might be a great way to push the, from a scientist viewpoint as well, to push some of these regulations forward, because I think it's quite clear now that sometimes, we continue doing the research, but sometimes if we're already aware of the regulations, we can do the research in a more focused manner, right? So I think that would be very useful. So I really, yes, Dr. Vishal.
Just one quick point. See, this is COVID time. We'll never get a better opportunity than this to actually talk to the world about it. So connect it with COVID and push the policy. Okay, thank you. I think those are very good insights for us as well. So we'll definitely move this forward. We actually have lots of ideas from while we were putting together the conference, and then just to see the general interest, I think this is a good time for CRISPR-Cas researchers to really have a voice. So I will end with that. Maybe Sudarshan, you want to add some more points?
No, I just want to thank all the panelists for their really, exciting discussion and all the aspects. You have highlighted everything from the scientific point of view. So from the organizers side, I would like to really thank all the panelists for taking time and giving your inputs. Thank you very much.
Thank you, and have a nice evening.
Have a nice evening.
Thank you. Thank you.
Thanks.
Thank you.
Thank you. Now we have just a general instruction for our audience attendees of the conference. So tomorrow we will start at 9:15 instead of 9:30. There was one talk that could not be entertained today because of technical difficulties. So we would like to have that talk come up front at 9:15. So please join us tomorrow, the last day of the conference, with as excitement as you're currently tomorrow. Okay.
Yeah, I'm assuming all of us who are still here are the hardcore conference people. So yeah. 9:15, we'll send you an email reminder as well, just to say that tomorrow it's going to start a little early. So yeah, thank you.
Thank you. Host, you can stop the